Good afternoon. So today we're going to cover protein structure, the basics of it. Now, this is something that a lot of people struggle to remember, and it's also absolutely essential for quite a lot of biology. So we're going to go over it relatively quickly. Now, I'm going to start off with amino acid structure. In all honesty, me telling you this is probably not going to help you to learn it at all. What you need to do is you need to go away and look, cover, write, repeat this. So if you don't know what I'm about to tell you, you need to almost pause me once I'm done telling you it, write it down yourself, and then literally just go away and just cover it, repeat it, cover it, write it, repeat it, and don't stop until you feel like you've memorized this structure, because it's one of the most commonly asked questions in all of biology. If you do feel like you know this, put up with this for 30 seconds. So all amino acids are the same in many ways. They all have a carbon in the middle, that's the first thing to say. Attached to that there is always four things. There are always four things. One of those four things is an amino group, NH2. You may sometimes see this written as NH3+. That's a chemistry thing. No one likes chemistry. It's one of those things. NH2 is fine. That's good. COOH. Again, you might see this as COO-. Again, chemistry. No one cares. Let's move on. This is the basics for biology. To be fair, if you wrote NH3+ or COO-, you wouldn't lose any marks. Let's just learn this one though. Then you've got that's called a carboxylic acid group. That's why it's called an amino acid. Amino acid. Attached to that, you always have a hydrogen on the bottom, and then there's an R group. Now, the R group can be literally anything, any chemical group whatsoever, and it will still be an amino acid. So, well, I'm not going to list things. Literally any chemical you could attach onto that. There are 20 types of amino acids found in your body, and by 20 types of amino acids, I just mean 20 of these with 20 different R groups. Again, if you haven't learned this, if you don't know this, go away. Just stop me. There's no point even carrying on until you know this. Because what I'm about to tell you is less important. Learn this. It's essential. So, if you have learned this, if you feel like you know it, let's go on to the more difficult bit. And this will mean me, unfortunately, skipping through a few slides, so excuse me while I do this, because this is just my teaching PowerPoint, and most of this is not useful to you at the moment. Right, there we go. Right. So, once you've got an amino acids on their own are pretty useless. They don't do anything at all. They're only useful when they're attached together to make proteins. Now, the first step of that is to make a dipeptide, and they'd expect you to be able to draw dipeptide out. Can you see these amino acids? Now, these are drawn in a slightly different way to a way you may have seen before. And I've done that deliberately because it's important for you to be able to recognize amino acids, even when they're laid out slightly oddly. You can see you've still got your carbon, your NH2, your COOH, your H and your R. They're just arranged slightly differently. Now, if you get this carboxylic acid group, this OH here, this hydroxyl group, this OH, and you get this amino group, this H here, you could get these H and this OH and you could make water. That would form what we call a condensation reaction, which I hope is a term you remember. And that would be what forms this dipeptide, and this is what this looks like. And it also makes water. Now, again, it's a really common question. How do you make a dipeptide? Well, you make a dipeptide by condensation reaction, joining together a hydrogen amino group and a hydroxyl group on a carboxylic acid. If you're not happy with hydroxyl group, just write OH. That's fine. Now, that's that. Again, little facts to know. All right? Now, this is called a peptide bond. A peptide bond. And again, a really common question. So there's now 3 facts we need to know. What's an amino acid look like? That's nothing I can teach you. You need to go away and learn it. What's a dipeptide made out of? People do get that wrong because they forget to write the second bit. They write, well, it's a condensation reaction. They forget to write it's a peptide bond. They forget to write it's made from the OH here and the H here. So, let's move on. The other thing, the main thing I wanted to focus on today was about protein hierarchy. And this is about how I go from some amino acids to an enzyme, a really complicated folded molecule with a really specific shape. And it's called, as I say, protein structure hierarchy. Now, the first step in this is to basically do this lots of times, to join together loads of amino acids between the carboxyl groups and the amino groups with condensation reactions to make peptide bonds. Now, Primary structure, the definition of this, the definition is the order, number, and type of amino acids in a protein. So I could get my 20 different amino acids. I could pick 6 of those. Those would be the types of amino acids I would have in my protein. I could get thousands of one and one of another. That's the number of different amino acids I have. And the order I put them in. So I can have 2 different proteins that are pretty much identical in every way. They have the same number of amino acids, they have the same type of amino acids. If I have them in a different order, there'll be a completely different protein, and it will have a different primary structure. So this word primary structure just refers to the order of the amino acids I have, the number of amino acids I have, and the type of amino acids I have. Now, if I have that, if I put that, if I say, well, I've got 30 glycines, 20 leucines, and 10 aspartamines, and I'm going to join them all together. I'm going to join them in this order. That's my primary structure. Now, again, at this point, this will be a useless thing. Just a chain of amino acids. Still won't do anything. Because in order for proteins to work, they have to have a particular folding. They have to be folded in a particular way. So, the first way they're folded is they're folded into their secondary structure. Now, the word secondary structure refers to 2 shapes. An alpha helix, which looks like this here, excuse the little box, and a beta-pleated sheet, which looks a little bit like this here. Really simple, small folds. And they're held together by hydrogen bonds between this hydrogen here and this oxygen here. Now listen, the idea of a hydrogen bond, I don't even want to delve into much right now. It's a really weak bond that holds together structures. We can talk about hydrogen bonds another time. As long as you understand that that is a hydrogen bond that's holding together, you can see there's a red dotted thing here, there's a red dotted thing here. It's holding together these really simple folds. So I get some amino acids, I get a particular order of them, a particular number of them, a particular type of them, and I make a primary chain. I fold them into an alpha helix, or a beta-pleated sheet, or a mixture of the 2. And that will be held together by hydrogen bonds. Still pretty useless though. This isn't specific enough to be an active site, or a channel protein, or anything. So now I need to fold them into a really specific shape. And that is... I don't want any of that, to be honest. Tertiary structure. Now, tertiary structure is where we get our proper shapes. Tertiary structure is where we're going to get complex, intricate folding. Now, in order for that to happen, we have R groups sticking out everywhere of my alpha helix, out of my beta-pleated sheet, out of my chain. Now, those R groups are going to attach to one another. They're going to bond to one another. Now, they might bond by hydrogen bonds. Remember, we said they're very weak. They might bond by things called disulfide bridges. And that's just a very strong bond between 2 sulfur-containing amino acids, for what it's worth. But you don't even need to know that. You just need to know the name, disulfide bridge. They could form by an ionic bond. Again, you should know what an ionic bond is from GCSE chemistry, but you don't actually need to. You just need to know that that's one of the ways we could fold this, is by getting 2 of the R groups, let's say one here, and forming a little ionic bond across them, so they get a little fold here. Or a hydrophobic-hydrophilic interaction. Now this is something I would like you to understand. The idea of hydrophobicity, is that a word? It's probably not. And hydrophilicness. The idea that some R groups will be water-loving, will want to be in the water, will be attracted to water, and some R groups will be water-hating, will be repelled by water. Now what will happen because of that is that some of the R groups will be stuck out on the outside, they'll be hydrophilic, because remember, the cytoplasm and the blood and tissue fluid, that's all water-containing. Some of them will be stuck in the middle, the hydrophobic ones, getting as far away from the cytoplasm, blood, tissue fluid as possible. And that's what enables things like enzymes to exist, because they have hydrophobic and hydrophilic R groups that stick out or in, creating this kind of globular structure. So far, I've got myself an order of amino acids, a number, a type, an order of amino acids. That's my primary structure. Because of that, they folded in particular ways because of hydrogen bonds between them to make this alpha helix and this beta-pleated sheet. Once I've done that, these alpha helices and beta pleated sheets fold themselves up again into even more complex shapes because of these 4 types of bonds. Now, most proteins just stop there. They have a primary structure, which is the order, number, and type of amino acids. They have a secondary structure, which is caused by the primary structure, the alpha helices, the beta pleated sheets, and they have a tertiary structure, the complex folding. Some proteins, though, need more than one polypeptide to do the job. Something like hemoglobin. Hemoglobin is made of 4 molecules, 2 beta molecules, 2 alpha molecules. The alphas are identical, the betas are identical. Now, the way in which these 4 interact and bond to one another will be determined by R groups that stick out of the alpha and beta chains. And they'll be kind of sticky to each other. They'll form little hydrogen bonds to one another. Or van der Waals forces will form if you're a chemist, which is another thing that can happen. And that forms a quaternary structure. Now, quaternary structure is just bonding between alpha and beta chains. No, it isn't. I apologise. Between any polypeptide. If you've got more than one polypeptide in an area and they bond to one another to do a job, they have a quaternary structure. And again, it's all about the R groups which stick out. Now, we'll learn about hemoglobin another time. We'll have a video about it, no doubt. That's not the point here. The point is that this is an example of quaternary structure. More than one protein bonding together. And again, this is all down to the primary structure, the secondary structure, and the tertiary structure, and the way in which these things are folded. That will then control the quaternary structure, how they're able to bind to other proteins. Now, at the end of every one of these videos, as you know, I have a little exam question. Now, I've made this one quite simple, in all honesty. A, B, C, 6 marks there. Have a look at that. Have a go at that. Because I would argue these are quite easy questions, because this is the start of year 12, effectively, this topic. I would expect you to be getting 5 or 6 out of 6 on this by the end of year 13. So have a go at this, see if you can get 6 out of 6. Think for each question, what could they possibly want me to write? Because the thing here will be that some people will miss out some points that are key. So pause me, have a go at this, and then we'll go through the mark scheme. Okay, hopefully you paused me. Right, describe how a peptide bond is formed. Condensation reaction, 1 mark. Always a condensation reaction. Between the amino group, NH2, and the carboxyl group, or the COOH. The end. Right? Now this second one will be the one people forget. You are expected to know instantly to accept amino and carboxylic acid group, because that's what I call them. That's one of the things you could call them. A lot of people forget the second bit because they forget what the groups are called, or they forget that it's NH2 and COOH. B. Well, what was it? The secondary structure of a polypeptide is produced by bonds between amino acids. Describe how. We're expected to know what type of bond it is. It's a hydrogen bond. I wouldn't expect you to know it's between the NH and the CO, though you might, because I vaguely mentioned it. The important bit you should be getting is that it's going to form beta pleat sheets and alpha helix. This is the thing that's really on the A level. So it's hydrogen bonds forming beta pleated sheets and alpha helices, because that's what secondary structure is. 2 proteins have the same number and type of amino acids but different tertiary structures. Explain why. Well, if they've got the same number and type of amino acids, well, we should know that the primary structure is the order, number, and type. So the thing that's left there is the order or the sequence. So you could say, well, they've got a different order of amino acids, different sequence of amino acids, forming ionic hydrogen disulfide bonds in different places. So the point with C here is, we're talking about if they've got the same number of amino acids and they've got the same type of amino acids, the only other thing that they can have different is that they will be ordered in a different direction. So instead of going lysine, glycine, glycine, it might be lysine, glycine, lysine. Different order. If it's a different order, they'll have R groups in different places. If it has R groups in different places, it will form bonds in different places. This is probably the hardest of the 3 questions. Now, if you didn't follow any of that, you need to come and see me. Just have a word. It's a 2-mark question. I'll be able to explain where you've gone wrong in 30 seconds, or any other biology teacher. If you feel like you want it explained in a different way, ask any biology teacher to explain to you the answer to this question. They'll be able to do it quickly. Okay, but hopefully that's all very clear to you. If, as I say, if it isn't, do ask me. Right, the end. Have a good day.